Learning outcomes. At the end of this lesson, the learner should be able to explain the role of genes in heredity, demonstrate one's understanding of key terms, and show in life situation genes' importance in education. What is genes?
What exactly is a gene? Each one of our cells contains 46 strands of DNA. A single strand is made of millions of particles called nucleotides, and these nucleotides come in 4 different types, which scientists have labeled A, C, T, and G. A gene is a special stretch of DNA, a sequence of A's, C's, T's, and G's that code for something.
Heredity. Heredity is the passing on of traits from parents to their offspring, either through asexual reproduction or sexual reproduction. The offspring cells or organisms acquire the genetic information of their parents. Through heredity, variations between individuals can accumulate and cause a species to evolve by natural selection. The study of heredity is biology of genetics. The role of genes in heredity. The heredity information is contained or within the genes, located in the chromosomes of each cell. An inherited trait can be determined by one or by many genes, and a single gene can influence more than one trait. A human cell contains many thousands of different genes in the nucleus. Heredity factors known as genes are thought to exist as discrete portions, known as loci, of chromosomes. The term discrete refers to the concept that genes are always located at the same point or locus on a chromosome. It is believed that pairs of homologous chromosomes contain linear, matching arrangements of genes exerting parallel control over the same traits. Pairs of genes that exercise such parallel control over the same traits are known as alleles, which are of 2 kinds, the dominant and recessive. Genetic characteristics can be extremely complex and may require the actions of several separate genes to be expressed in the adult organism. An example of traits that require more than a single pair of genes to be expressed are human height and human color. Other traits may be controlled by a single pair of genes. Some of these traits may have extreme phenotypes, such as albinism, lack of sin segment in humans. There are common genetic disorders. The first one is Down syndrome. Down syndrome is a condition in which a person has an extra chromosome. Chromosomes are small packages of genes in the body. They determine how a baby's body forms and functions as it grows during pregnancy and after birth. Typically, a baby is born with 46 chromosomes. The second is sickle cell disease. Sickle cell disease is a blood disease that affects red blood cells. Normal blood cells are round, and people with sickle cell anemia, hemoglobin, a substance in red blood cells, becomes defective and causes the red blood cells to change shape. The third is phenylketonuria or PKU. Is a rare but potentially serious inherited disorder. Our bodies break down the protein in foods such as meat and fish into amino acids, which are the building blocks of protein. These amino acids are then used to make our own proteins. The last one is hemophilia. Hemophilia is usually an inherited bleeding disorder in which the blood does not clot properly. This can lead to spontaneous bleeding as well as bleeding following injuries or surgery. Blood contains many proteins called clotting factors that can help to stop bleeding. There are types of hemophilia. The first one is hemophilia A. Most common type of hemophilia: 8 out of 10 people with hemophilia have hemophilia A. People with hemophilia A do not have enough clotting factor 8. The next is hemophilia B. It's also known as Christmas disease and a least common type of hemophilia. People with hemophilia B do not have enough clotting factor 9. It is caused by a deficiency in clotting factor 9. The last is hemophilia C. It's also called deficiency of factor 11, a mild form of hemophilia. It is inherited differently than hemophilia A or B. As a result, it can be passed to both male and female children. Each of our gene codes contains unique protein, and each protein has a separate and distinct function to perform in the cell. Effects of genes in our environment. Genes are inherited, but their expression can be modified by interaction with the environment. Genetic traits are determined largely through the precise information found in the cell's gene structure. A variety of factors in the environment can actually alter the effects of a particular gene. Some examples of this effect are as follows. Effect of light or chlorophyll production. Although most plants have the genetic ability to produce chlorophyll, they will do this only in the presence of light. Without light, these plants produce only a light yellow pigment and therefore appear pale and sickly until they are exposed to sunlight. After a few days of exposure to sunlight, the chlorophyll reproduction mechanism is enabled and green color returns. Cells store and use coded information. The genetic information stored in DNA is used to direct the synthesis of the thousands of proteins that each cell requires. The cell contains many thousands of such codes in its chromosomes. Each strand of DNA in a chromosome has the potential to provide the complete chemical code for the manufacture of at least one complete protein. These proteins are highly specific and they result in the expression of some specific trait or portion of a trait in the living cell and consequently in the organism of which they are part.
So good day everyone, I'm Amika and I discuss about the gene mutation. So what is gene mutation? Gene mutation are segments of DNA molecules. Any alteration of DNA sequence is a mutation. Usually, an individual cell's altered gene will be passed into every cell that develops from it. So gene mutation may be defined as being any changes in the nitrogenous base sequence of a molecule DNA. When the base sequence of DNA is altered, the amino acid sequence of the polypeptide for which is code will likewise be altered. Such an alteration may affect the operation of the resulting enzyme, preventing it from properly catalyzing its reaction and thus preventing a trait from being expressed by the cell. The majority of gene mutations are harmful because they result in the cell being impaired from performing some specific task. In rare cases, a mutation may result in a lethal gene that kills the cell either by proceeding a substance toxic to the cell or by failing to produce a protein or vital important to the cell. So gene mutations are passed to every cell that arises from the mutated. If the mutation occurs in somatic tissue, a defect is limited to the tissues immediately surrounding the mutated cell. If the mutation occurs in a primary sex cell, it passes onto the offspring that result from fertilization of or by gametes produced from the primary sex cell. A mutation can enter the gene pool of a population and be passed on the succeeding generation. This is known to be a cause of vibration in the species. On the other hand, DNA is a polymer made of repeating chemical unit known as the nucleotide. Thousands of units are known to comprise a single DNA molecule, making it one of the largest of all organic compounds. DNA exists in hundreds of thousands, if not millions, of different forms. Depending on the precise arrangement of nucleotides in the molecule, there is variability is the key to genetic variation in living things. So DNA nucleotides themselves are quite complex, being composed of 3 separate subunits. So the first is phosphate group. Phosphate group, a chemical group made of phosphorus and oxygen. A phosphate group, a functional group or radical, comprised of phosphorus attached to 4 oxygen. So the second is deoxyribose. Deoxyribose, a 5-carbon sugar made of carbon, oxygen, and hydrogen. The deoxyribose, also known as deoxyribose and 2-deoxyribose, is a pentose sugar or monosaccharide containing 5 carbon atoms. That is a key component of nucleic acid, deoxyribonucleic acid. Deoxyribose is the sugar component of DNA. Just a ribose serve that role in RNA ribonucleic acid. So the last is nucleobases. Again, the last is nitrogenous base. A nitrogenous base is a molecule that contains nitrogen and has the chemical properties of a base. The nitrogenous base in the DNA are adenine A, guanine G, thymine T, and cytosine C. The nitrogenous bases or RNA are the same, with one exception: adenine, guanine, uracil, and cytosine.
DNA is perhaps the most famous biological molecule. It is present in all forms of life on Earth. But what is DNA, or deoxyribonucleic acid? Here, we cover the essentials. Virtually every cell in your body contains DNA, or the genetic code that makes you. DNA carries the instructions for the development, growth, reproduction, and functioning of all life. Differences in the genetic code are the reason why one person has blue eyes rather than brown, why some people are susceptible to certain diseases, why giraffes have long necks. Amazingly, if all of DNA in the human body was underbuilt, it would reach to the sun and back more than 300 times. In this article, we break down the basics of the DNA, what it is made of, and how it works. What is DNA? In short, DNA is a long molecule that contains each person's unique genetic code. It holds the instructions for building the proteins that are essential for our bodies to function. DNA instructions are passed from parent to child, with roughly half of child's DNA originating from the father and half from the mother. Structure. DNA is a two-stranded molecule that appears twisted, giving it a unique shape referred to as the double helix. Each of the two strands is a long sequence of nucleotides or individual unit made of a phosphate molecule, a sugar molecule called deoxyribose containing 5 carbons, a nitrogen containing region,
There are 4 types of nitrogen containing regions called bases: adenine, cytosine, guanine, and thymine. The order of the 4 bases forms the genetic code, which is our instructions for life. The bases of the 2 strands of DNA are stuck together to create a ladder-like shape. Within the ladder, A always sticks to T and G always sticks to C to create the rungs. The length of the ladder is formed by sugar and phosphate groups, packaging DNA, chromatin, and chromosomes. Most DNA lives in the nucleic of cells, and some is found in mitochondria, which are the powerhouses of the cell. Because we have so much DNA, and our nuclei are so small, DNA has to be packaged incredibly neatly. The strands of DNA are looped, coiled, and wrapped around proteins called histons. In this coiled state, it is called chromatin. Chromatin is further condensed through a process called supercoiling, and it is packaged into structures called chromosomes. These chromosomes form the familiar X shape as seen in the image above. Each chromosome contains one DNA molecule. Humans have 23 pairs of chromosomes, or 46 chromosomes in total, interestingly. Fruit flies have 8 chromosomes, and penguins have 80. Chromosomes 1 is the largest and contains around 8,000 genes. The smallest is chromosomes 21 with around 3,000 genes.
The genetic code. Every organism requires a set of coded instructions for specifying its traits. For offspring to resemble their parents, information must be reliably transferred from one generation to the next. Heredity is the passage of these instructions from one generation to another. Discoveries in genetic science, deoxyribonucleic acid or DNA was revealed to be the chemically active agent of the gene. DNA replicates itself when chromosomes replicate in the early stage of cell division. DNA is passed from generation to generation during reproduction and acts as genetic factors. DNA interacts with the cell's chemical factory and produces the observable effects of the phenotype when genes are inherited by a cell or an organism. DNA regulates the productions of enzymes in the cell and thereby enables the cell to perform the complex cellular chemical reactions necessary to sustain life. Stem cell therapy focuses on the use of stem cells to treat or prevent a disease or condition. Stem cells are a special red of cells that are capable of differentiating into more than one kind of tissue. Stem cells are used for curing various diseases such as diabetes, arthritis, few cancers, bone marrow failure. Genetic engineering is a technique of controlled manipulations of genes to change the genetic makeup of cells and more genes across species boundaries.